Good morning everyone. I'll be dealing with randomized control trial in today's class. As we see in the picture, this is a particular study, epidemiological study design, and all the factors mentioned here gives an advantages of randomized control trial. So the randomized control trial is one of the epidemiological study design. It has greatly contributed to the advancement of the research in scientific medicine. Randomized control trial is a type of epidemiological study design which is highly reliable and most accurately tests the research hypothesis. So among the several research design, the randomized control trial is accurately predicting the research hypothesis, and randomized control trial is widely recognized as a gold standard for evaluating the healthcare technologies. So if you carefully look into the various epidemiological methods, the epidemiological methods are broadly classified into two broad categories. The one is observational studies and the other one is experimental studies. Observational studies helps in formulating and testing the hypothesis, whereas the experimental studies helps in proving the hypothesis. We need the scientific proof for the etiology of the disease. That is only possible through experimental or interventional studies. So there is a scope for error in case of observational studies, whereas the data is more accurate and more reliable in case of experimental studies. So if we carefully look into the various types of research designs, the observational studies at a lower level of evidence-based medicine and the randomized control trials at a superior level at the higher level of evidence-based. The randomized control trials are further contributing to the systematic review and meta-analysis, which is considered as the highest level of evidence in evidence-based practice. So whenever we are providing evidence for the clinical practice, we need to take the help of research, and the systematic review with a meta-analysis where the randomized control trial is widely used is regarded as the highest level of evidence. So we are discussing the randomized control trial under the heading of experimental studies. The experimental studies are basically used to provide the scientific proof for the etiological factors which may permit the modification or the control of those factors, to provide the method of measuring the effectiveness and efficiency of health service for the prevention, control and treatment of disease, and improve the health of community. These are the two broad uses of experimental epidemiology. The first use is to provide the scientific proof for the etiology or the risk factors, and to provide the measure for measuring the effectiveness and efficiency of various health services for the prevention and control of disease. So prior to conduct the experimental studies on human, animal studies are very much required. The more important application of animal experiments have been in experimental reproduction of human disease in animals to confirm etiological hypothesis and to study the pathogenesis phenomenon or mechanisms. Testing the effectiveness of various preventive and therapeutic measures such as vaccines and drugs, and completing the natural history of the disease. These are the three important features of animal studies where we are going to use it for confirm the etiological hypothesis, we are using it for testing the efficacy of various preventive and therapeutic materials, and we are studying the natural history of the disease with the help of animal studies. The advantage of animal studies are the experiment animals can be bred in the laboratories and manipulated easily according to the wishes of investigator. They multiply rapidly and enable the carry out certain experiments which in human population would take several years and involve many years, many generations. These are the advantages of animal experiments, whereas the limitations or the disadvantages include all human disease cannot be reproduced in animals. And the conclusion derived from the animal experiment may not be strictly applicable to human beings. Only with certain degree of accuracy we can extrapolate to the human beings, but not all the disease because of some genetic inherent problems. So human experiment will always be needed to investigate the disease etiology and to evaluate the preventive and therapeutic measures. These studies are even more essential in the investigation of disease that cannot be reproduced in animals. Whenever the studies cannot be possible in animals, the human studies become inevitable and they serve as the best level of evidence. Randomized control trials are quantitative, comparative controlled experiment in which the investigator study two or more interventions in a series of individuals who receive them in random order. RCT is one of the simplest and most powerful tool in clinical research. The basic steps in conducting a randomized control trial include drawing up of a protocol, selecting a reference or experimental population, randomization, manipulation or intervention, follow-up and assessment of the outcome. So this is a further elaboration of the same thing. When you are taking a reference population, that is, you have to select the suitable population from which you are drawing a experimental or study population. After eligibility criteria, we are randomizing and followed by manipulation, follow-up and assessment of outcome. We are going to discuss these steps in detail. The first step in randomized control trial is drawing up of a protocol. This is like a blueprint. Before conducting a randomized control trial, we should have a proper protocol. The protocol should explain about the aims and objective of the study, the question to be answered, that is the research question. We have to be explicit about the research question or research hypothesis, the criteria of the selection of the subject. The protocol should mention the inclusion and exclusion criteria properly. The size of the sample, how many subjects are going to be included, it should be exclusively mentioned. The procedure for allocation of subject into study and control groups, that is how the method of allocation, whether the type of concealment has to be clearly explained, and what groups are receiving what kind of treatment. The treatment to be applied in each group has to be explicitly mentioned. And when and where and to how to and what kind of patients, standardization of working procedures. So all this has to be mentioned well in advance in the protocol, so that throughout when you are conducting a study, we should be adhered to all these protocols. The protocol aims at preventing bias and to reduce the source of error in the study. Whenever you are conducting a multicentric trial, so the protocol helps in maintaining the uniform standard for conducting the randomized control trial. Selecting a reference and experimental population. So the first step in randomized control trial is selection of a reference population. It is a population to which the finding of the trial, if found successfully, is expected to be applicable. That is a drug, vaccine or any other procedures. So this means to say that whenever we are choosing a population, the study result should be made applicable to that population. The reference population can be as broad as a mankind, or it should be confined to certain geographic area that includes a school population, industry or some specified occupation. So the next comes the experimental or the study population. The study population is derived from the reference population. It is the actual population that the participate in the experimental study. Ideally, it should be randomly chosen from the reference population. The participant or the volunteer must fulfill the following 3 criteria. The experimental population is chosen from the reference population. Ideally, it should be randomly chosen, and they should be able to fulfill the following criteria, such as informed consent. They should be able to agree to participate in the study. They should be representative of the population to which they belong. They must be closely resemble, that they must be very much similar to the parent population or the reference population. So as I said, there are 3 important criteria, that is informed consent, they should be representative of the population, and they should be able to fulfill the eligibility criteria. The patient should be selected based upon the inclusion and exclusion criteria. So as you can see in the previous diagram here, so this is the target or the reference population from which the sample is selected. So this is called the sampling frame. From the reference population, the sampling frame is decided. From the sampling frame, the sample is chosen. So it should be in such a way that the sample is representative of the target population. Randomization, it is a statistical procedure by which the participants are allocated into groups usually called as the study and control group. Randomization is an attempt to eliminate the bias and allows for the comparability. Randomization is the heart of randomized control trial. Randomization ensures that the investigator doesn't have the control over allocation of participant into study and control groups. Every individual get an equal chance of being selected into the either group with the help of randomization. The randomization is considered as one of the key steps in randomized control trial. It gives the equal chance for every individual to be selected, and it ensures that the investigator doesn't have the control over allocation of participant into study and control group. So this is an example of the randomization is achieved by the table of random numbers, or it can be used by a table of lottery or dip method, or you can use a toss a coin or flip a coin, or you can use a dice. Based on the dice, we can allocate the subject into study and control groups. Suppose if you are using a total number of random number table, now if you are requiring around 200 subjects for your study, you are randomly choosing one number from the table. Imagine that you are selecting 269 here. So the 269 is the first number, and you are choosing a number below 200 from the below lines in particular column. If suppose you are choosing a 269 here, 269 is above than 200, so you are taking the values below 200. That is 128 is the first number, 620 is higher, we are not selecting. 013, so the 120 is the first subject, 013 is the second subject, 001 is the third subject, 006 is the fourth subject. Like this we are selecting the each individual from the random numbers, and they are going to be our sampling units. So other method is flip a coin. So you are flipping a coin, those who are getting a head will be allocated into group A, and those who are getting a tail, they will be allocated into group B. In the same way the dice can be used. So when there is an odd number, the subjects are allocated into group A. If it is an even number, the subjects are allocated into group B. So the next important step in randomized control trial is manipulation. This is also called intervention or experimentation. This is the deliberate application or withdrawal or reduction of the suspected causal factor. This may be a drug, this may be a vaccine, this may be a dietary component, habit as laid down in the protocol. The manipulation creates an independent variables whose effect is then determined by measurement of final outcome, which constitute dependent variable. When you try to intervene, it creates a dependent variable and independent variable. So the effect of independent variable is tested on dependent variable. So after intervention, the subjects are allocated into two groups. There is a one comparison group and there is a one study group. So both the subjects are followed up for a defined time period. This is called follow-up. This implies the examination of the experimental and control group subjects at a defined intervals of time in a standard manner. The group should be observed with equal intensity under the same given circumstances in the same time frame till the final assessment of the outcome. The subjects in both the groups should be observed in the same intensity throughout the study period. The follow-up may be short or may require many years depending upon the study undertaken. The follow-up duration should be carefully planned based upon the nature of the disease, based upon the objective of the study. It could be 3 months, it could be 6 months, or it could be 1 year. So based upon the objective of the study, based on the outcome and its half-life, you are going to decide the follow-up time period. So one of the important drawback of randomized control trial is the loss to follow-up. Some losses to follow-up are inevitable, such as migration, loss of interest, or the patient might die in the course of studying. So that could affect the study results. So you have to adjust for follow-up. There is something called intention to treat analysis. You have to assume that these subjects are going to become a part of the study, and assuming that they have participated and the data is treated as intention to treat with the intention to treat analysis. The assessment of outcome. So the last important step in randomized control trial is the assessment of outcome. The study is evaluated for final outcome. So the result could be a positive result or the study could be negative results. Positive result, that is benefits of the experimental measures such as reduced incidence or the severity of the disease. Cost to the health service or other appropriate outcome in the study and control groups. Either there will be improvement in the disease process or there is a reduced incidence of the disease, that could be termed as a positive effect. Negative results in terms of the severity and the frequency of side effects, complication including death. The study may show negative effect in terms of what I can say, like in terms of complications or in terms of any side effect, or there may not be any improvement as expected. So that can be termed as a negative results. So there is a scope for error in randomized control trial. That could be due to participant, that could be due to subject or participant, that could be from the part of investigator or the researcher, or that could be from the person who is evaluating or analyzing a data. So all these kinds of error can be eliminated by a procedure called as a blinding. So single blind trial, the trial is so planned that the participant is not aware whether he belongs to the study group or the control group. Whereas in double blind trial, the trial is so planned that neither the doctor nor the participant is aware of the group allocation and treatment received. In single blind trial, either the participant or the participant is not aware of the group allocation, whereas in case of double blind, both the participant and the researcher is not aware about the group allocation or the treatment they are receiving. In triple blind trial, this goes one step further. The participant, the investigator and the person analyzing the data, all three are blinded. So this is highly reliable and the most accurate, but however because of some practical reasons triple blinding is not possible all the time. So most of the time the researcher do confine to double blind control trials, but whenever the provision is given, one should be aiming at triple blind trial. So among the several other designs, the variation of the randomized control trial. So there are two important variations in the study design. The one is called as a parallel study design and the crossover study design. This is also called as a concurrent parallel design. In concurrent parallel design, there are two groups. Throughout the study period, the study and control groups are followed up over a time period, and they will be remain in the study and control group throughout the study period. There is no interchange or there is no exchange of group allocation. Whereas in crossover study, there is two groups. For a certain time period in the beginning, both the groups are continued in each groups. After a certain time period, the group B is converted into group A and the group A is converted into group B. You are giving a definite washout period in between. So this washout period is depending upon the drug half-life or the duration of the disease. Based upon those characteristics, you are defining that washout period. You are completely allowing the carryover effect, and then you are inter-exchanging the group. So the advantage of this crossover study design is that the subjects are each acted in both the groups. So any possible errors in each sample could be overcome by this particular study design. So you can see here the crossover study design. There is two groups, and after certain washout period, the control group is used in a study sample, whereas the intervention group is changed into control group, and you are evaluating the result after a defined time period. So moving on to the other types of randomized control trial. There are various other types of randomized control trials. They are categorized into clinical trials, preventive trials, risk factor trial, trial of etiological agents, SAJJAN experiment, and evaluation of health services. For the most part of clinical trials have been concerned with evaluating the therapeutic agents, mainly drugs. It includes the evaluation of beta blockers in the reduction of cardiovascular mortality in patients surviving the acute phase of myocardial infarctions. So here the clinical trials are refers to evaluation of certain therapeutic drugs or therapeutic procedures, or it could be some therapeutic materials as well. So many ethical, administrative and technical problems are involved in the conduct of clinical trials. The major problem with the clinical trial is the ethical issues. We have to take the safety into the consideration, or the ethical issues into consideration while conducting the clinical trials. There are various phases of clinical trial. These are including the preclinical studies, the phase 1, phase 2, phase 3 and phase 4. The preclinical or laboratory studies are very much essential when you are conducting randomized control trial. It rules out the toxicity, so then it can be safely carried out in human beings. Prior to conducting human experiments, the animal experiments are very much required, and in vitro studies also has to be conducted to give the preliminary evidence for human studies. In phase 1, we are looking for the evaluation of the safety. Predominantly, we are looking for the safety, and we gather the information about how a drug interacts in the human body. This study we are conducting on a very small number of subjects, that is around 15 to 20, and predominantly we are looking for a safety. Whereas in case of phase 2, we are looking for predominantly safety and partly the efficacy. Roughly around 80 to 100 subjects are being evaluated. It is to further evaluate the safety, monitor the side effect, check which dose work best, and check the effectiveness. Whereas in case of phase 3 trials, we are predominantly looking for the safety and efficacy. Predominantly efficacy, partly safety, to confirm the effectiveness and monitor the safety. So the subjects include up to 300 to 3000 populations. So in case of phase 4, we are gather information on the drugs effect in various populations. So when the trial is being tested among different population across the different geographic area, and also any side effects which are associated with the long-term use. When the drug is released into the market, and when the drug is used for a long time in the market, we are looking for the long-term side effect. So the preventive trial is another type of clinical trial where it implies the trials of primary preventive measures. So this includes the trials use of vaccines and chemoprophylactic drugs. So in the dental field, we can tell about the use of various mouthwashes, dentifrices, or any fluoride varnish. So predominantly we are focusing on preventive materials, the use of dentifrices in controlling the plaque, the use of mouthwashes in controlling gingivitis. So these kind of examples where instead of a drug you are using some preventive material, hence these are called as a preventive trials. So risk factor trial. A type of preventive trial is a trial of risk factors in which the investigator intervenes to interrupt the usual sequence in the development of disease for those individuals who have risk factors for developing a disease. Often this involves risk factor modification. The concept of risk factor gives a new dimension to the epidemiological research. So instead of a drug, instead of any preventive material, you are testing how far a risk factor is associated with a disease. It could be a multiple risk factor or it could be a single factor. So you are looking for the possible relation or the association. To confirm that association between risk factor and disease, you are using risk factor trials. The SAJJAN experiments. In this type of study, an attempt is made to evaluate the termination of habit, which is considered to be causally related to a disease. So in this kind of SAJJAN experiment, one group is continuing with the continuation of the habit, the other group is going to reduce the habit, and over a certain period of time you are going to see where there is a improvement in the condition among the group which has stopped the habit and which has continued the habit. The good example is the smoking habit. If such action is followed by significant reduction in the disease, the hypothesis of cause is greatly strengthened. The familiar example is cigarette smoking and lung cancer. As I said, the one group is continue to smoke and the other group is stopping smoking, and you are going to see whether there is a reduction in the incidence of lung cancer or not. The trial of etiological agent. One of the aim of experimental epidemiology is to confirm or refute an etiological hypothesis. So we discussed about a risk factor, we discussed about the preventive trial, we discussed about the clinical trial. Now there is a one definitive causative factors, and what is its relation to the disease process. So since most diseases are fatal, the disabling or unpleasant, human experiment to confirm an etiological hypothesis are rarely possible. We cannot conduct most of the experimental epidemiology for causal hypothesis because of the safety or ethical reasons, because of possible risk to the human beings, possible risk of any kind of complications or the death. We are not supposed to conduct any kind of trial of etiological agents are rarely used to confirm the etiological hypothesis. Evaluation of health service. Apart from this, randomized control trials have been extended to assess the effectiveness and efficiency of health service. The community-based programs, when they are being used, the success or failure of the program can be used by the method of randomized control trial. This is also another type of randomized control trials. So in comparison to randomized control trial, the other type of study designs are non-randomized trials. Although the experimental method is almost always to be preferred, it is not always possible for ethical and administrative and other reasons. So because of safety issues or because of some administrative reasons, you will not be able to conduct randomized control trials. In such conditions, some non-randomized trials are used. These are called uncontrolled trial, natural experiment, and before and after comparison studies. However, even in these uncontrolled trials, one is using implied historical controls, that is the experience of earlier untreated patient affected by the same disease. There is a room for uncontrolled trials, trials with no comparison groups. Uncontrolled trial may be useful in evaluating whether a specific therapy appear to have any value in a particular disease, to determine appropriating the doses or investigating the adverse reactions, etc. The natural experiment, where experimental studies are not possible in human population, the epidemiologists seek to identify natural circumstances that mimic an experiment. In respect of cigarette smoking, the people have separated themselves naturally into two groups, smokers and non-smokers. So while you are conducting the randomized control trial, some of the ethical issues are very much important. So the first thing is beneficence, which require that definite good out of the trial. So the researcher should ensure that there must be a definite positive outcome of the research. There is no point in unnecessarily conduct the research whenever it is not needed. and the researcher should also ensure that there is no sufficient harm to the research participant. This should be used by proper conducting phase 0, phase 1 trials, and also the animal experiment and preclinical studies has to be conducted well in advance. Respect for person. Whenever you are recruiting the research participant, we should ensure that there is a free choice for the participation. The participant should not be enforced. The participant should not be forced to be part of the research trials, and whatever the research information they are revealed by the research participants should be kept confidential. It should not be revealed until and unless informed to the participants. So all these factors should be given a due importance whenever you are conducting randomized control trial. So overall, the randomized control trials was one of the best study designs, which is highly accurate, most accurate, and it serves as the best level of evidence-based medicine. Thank you.